Hello everyone, I'm Weilong from China Agricultural University. I'm glad to have the opportunity to introduce our recent work on the topic of network-oriented strategies to connect the assemblies and the germplasm resources in Triticeae. For model species Arabidopsis, it takes about 20 years for the genomic research to go through from the first high-quality genome assembly to have a map to then large-scale population sequencing and resequencing and multiple assemblies or pan-genome, and very recently to the gap-free genome. For rice to go through such process, it takes 50 years. For maize, it takes 12 years. As wheat is a polyploid species with huge genome size, almost the 40 times of rice genome and the 6 times of maize genome. It takes only 2 years for wheat community suddenly enter the post-genomic and pan-genomic era with the burst in both assemblies and resequencing data. For Triticeae tribe, 67 assemblies have been published in total. That is 116 deployed assemblies. Especially 80 new assemblies were just published since last year. Our group in China Agricultural University have also contributed a de novo assembly of the Tibetan semi-wide wheat Zhang 1817, which is published on Nature Communication in 2020. This genome is the second formally published chromosome-level assemblies of hexaploid wheat. Here we faced with such question: how to deal with the emerging assembly in Triticeae tribe species, and how to efficiently explore the homologous genes at pangenomic level. Actually, the homolog inference problem is never a trivial task, especially for species in Triticeae tribe, as these species generally harbor huge genomes and have complex relationships as a result of reticulate evolution and with frequent polyploidization and regression. We'd like to introduce our work, which has been published on 2020, as aiming to establish the global homolog inference method GPF. Additionally, a homolog database for developed, which has been currently widely used, especially by the community. First, we organize the assemblies in Triticeae hierarchically at 6 levels. The homologs were specifically inferred between the subgenomes. Additionally, we deal with the polyploid genomes by decomposing the task into multiple subtasks of homology inference between diploid subgenomes. One advantage in our method is to incorporate the quality information for homolog inference, which quite significantly improves the inferred result for closely related genomes. Another advantage is that we propose the 4 different types of homolog relationship include RBH, SBH, one-to-many, and singleton relationship. And these relationships between two accessions are directional for query-targeted pairs. This is a flowchart illustrating the design of GenTribe, which integrates sequence similarity and collinearity scores with a dynamic parameter selection strategy, and finally constructed multiple homolog gene tables and collinearity block tables. These generated tables serve as the basis for the database construction and suppose further functions of homology query, analyzing, and visualization. A systematically evaluation on the homolog inference performance have been done by comparing with other widely used tools and databases. Our method GenTribe has the highest records with comparable accuracy. More actual homolog pairs were inferred with support of collinearity and sequences similarity ratios. A case will show here that red lines indicate that sure homology pairs inferred by GenTribe but failed by other tools. Then we developed the TGT database as an online server supporting the exploration of homology genes in Triticeae tribe, and incorporated with functions of homology query analysis and GO analysis. One can just click the buttons on the web pages and generate the tables and the figures with user-friendly interface and versatile parameter. Remarkably, we implemented the collinearity analysis and visualization function for both microcliniarity and the macrocliniarity. We constructed the cascaded profiles of microcliniarity chromosome blocks at the chromosome levels. By tracing the blocks of a subgenome in Chinese string, we can see that the Asian interchromosome translocation that connected the chromosome 4A, 5A, and chromosome group 7. We, we constructed we were able to present a precise picture delineating the rearranged region for the weight 4A, 5A, 7B chromosome translocation at a single gene level. Finally, we proposed a refined model describing the 4A, 5A, 7B translocation by two main translocation events. The first events occurred before the formation of urarial with the diploid progenitor of a lineage. The long arm ends were chained between the 4A and 5A. The second translocation events occur during the polyploidization process when forming the tetraploid with the triticum dicocoides. The blue block moved to the short arm end by dragging a piece of the red blocks and recruit another purple piece from the short arm of 7B. Then the previous short arm of 4A became the new long arms. For microclinical analysis, we can trace the dynamics of neighboring gene contacts with cascaded homology relationship from the weight to the body, and then even to the rise and to the maze. We also illustrate the complex evolutionary history of the floral repressor gene, VRN2, which involved as a combined result of genome translocation, duplication, polyploidization, and gene loss events. Here is a short summary for this part that we successfully connect the emerging assemblies in Triticeae by genes in a homology network. As another aspect, hundreds of beta sessions with resequenced data were published since 2020. Here comes the question, how to explore and utilize these published results. In 2020, we have a paper published on GigaScience, which is describing a novel web server framework named SMB Hub. This tool can be utilized to quickly set up a web server for exploring the larger scale genomic variation data. Generally, SMB Hub supports queries, analysis, visualization for SNPs at haplotypes. Tables and high-quality figures can be downloaded directly. A portal for multiple SMB servers were constructed named with SMB Hub portal, which currently covered around 3,000 sessions as a full collection of the published genomic variation data in Triticum. Here comes to another question. What could the sequencing data tell us about the relationship in genomic regions that were shared or differed among these wheat varieties? We recently published a paper on plant physiology describing a novel method for dissecting the germplasm resource in genomic regions. For developing these novel methods, we take advantage of the observations on the distribution of the stratified SNP densities by comparing the two with cultivars. Then we developed the ggComp algorithm by integrating SNPs and CMV blocks identified by resequencing data, and also combined with the EM algorithm and the hidden Markov model. The ggComp method could identify germplasm resource-based IBD blocks at the 1 megabyte resolution for a pair of wheat varieties. Finally, PHR and SGR regions and several CMV regions will be inferred. This is an example as revealed by ggComp when comparing two siblings, BIMA1, BIMA4. Light blue region indicates a shared germplasm resource-based IBD between two varieties, and the dark blue indicates the genomic regions descended from different backgrounds. With ggComp, we were able to profile genomic regions of a modern cultivar Nomda 5181 as descended from its parents. A total of 91 breakpoints were found, which corresponds to 4.3 breakpoints per chromosome, indicating very limited crossovers in its breeding practice. For a collection of 212 wheat accessions, we pairwise compared these accessions and were able to build a wheat genomic-based germplasm network notated as ggNet. We can see how these modern wheat cultivars are connected and understand the hidden relationships in network. For example, several key exotic lines contributed a high proportion to Chinese modern wheat cultivars, while most Chinese landraces were actually underexploited in modern wheat gene pool. Moreover, we could also visualize the network for local region of specific genes, such as the Green Revolution gene RHTB1 and RTD1. We can see the cluster of accessions, which actually corresponded to the diverse haplotype at the level of germplasm resource. Thus, we named each cluster as G-hub. We could see the largest G-hub is actually the semi-developed alleles RHTB1b and RHTD1b. Excitingly, this inferred G-hub perfectly fit with previous identified alleles for the two genes. And we also find two previously unreported alleles, which is under-selected for RTD1. Thus, utilizing ggComp, we were able to compare any pair of accession for inferring GIBDs without prior information on recorded pedigree. And we can construct a multiple scale ggNet at the whole genome level, at the chromosome level, and at the local region level. We finally developed a database named WheatCompDB for presenting the genetic relationship for the wheat germplasm resources. Here's an animation example for using WheatCompDB to compare any accession and to visualize the GIBD on the chromosome is annotated. A brief summary for this part, we developed a novel method ggComp for dissecting GIBD blocks, and then were used to connect the germplasm resource in wheat. These are all the works presented today, which cover in 4 main papers that we published in the last 3 years. Here's a graphical summary presenting novel algorithms we developed in recent years and the database we constructed for wheat field, and the new biological findings were highlighted based on these tools and data resources. We thank all the colleagues, students, collaborators for their important contributions.